Good evening, everyone, and welcome to this fabulous venue for what I know is going to be a really fascinating talk. So I'm Helen McShane. I'm a professor of vaccinology at the University of Oxford, and I'm a co-director of the Validate Network, together with our guest speaker, Adelaida. Validate is a vaccine network formed in 2017, and we focus on three groups of pathogens or bugs. So we focus on mycobacteria, which cause TB and leprosy. We focus on Leishmaniasis, of which more shortly, and we focus on melioidosis, which is a disease particularly endemic in Southeast Asia. We focus on all of these pathogens because they cause a huge amount of disease, particularly in low and middle income settings, and they are complex, which is my euphemism for difficult, to make vaccines for. So that's why we're focusing on these pathogens. What Validate does is it provides pump priming funding, it provides fellowship funding, travel and training grants, it provides annual meetings, mentoring, and a network, particularly for early career researchers and the next generation of biomedical scientists, to encourage them to work on these difficult pathogens. We've gone from strength to strength since we were first formed in 2017. So we had 32 founding members in 2017. We've now got over 1,000 members who are all biomedical scientists working in this field across 400 institutions in about 79 countries around the world. It is my great pleasure this evening to introduce Dr. María Adelaida Gómez, who is a leading biomedical scientist in Colombia, who has spent her career working on Leishmaniasis in humans to try and understand why some people get very sick with Leishmaniasis and some people don't. She's done a huge amount of outreach work, particularly in indigenous communities and in rural areas, and she's funded by the NIH and Wellcome, amongst others. So Adelaida, welcome and thank you.
All right. Woof. Here we go. I guess you can hear me well now. Good evening, everyone. Thank you so very much for being here. It is a very huge pleasure for me to be standing here talking to you today about my favorite parasite that causes a disease that is very complicated and complex, as Helen was saying. And I was asked just before we started, why do I still have hope? And the hope is seeing people like you coming to this type of events. This gives me a lot of hope that a lot can be done and that the word is going to get out there of the need and the importance of these infectious diseases that we work with and that we devote our careers working through. So, you're going to see a lot of pictures. Every single picture besides or except the pictures from patients have been taken by me in different field trips related to the work that we do. So mind the quality, some may not be amazing, but I'm bringing them because I want to bring you through the journey of investigators and patients through Leishmaniasis research and also through what patients have to go through when they are seeking care. The pictures depict very beautiful areas of Colombia, and this is the reality of our country. So I want to kill two birds with one stone. One, get you in love with the country where I come from and hopefully get you to visit one day. And two, see the reality of patients in these areas. Despite the beauty of nature in these places, it is very tough to live in some of the rural areas that I'm going to show you. So you're going to see in the top of each slide the place where it has been taken. So this one, for example, was a field trip that we were doing to an island in the Pacific area of the country, and it's an area called Sala Honda. So you're going to see the references, the geographical references there. So again, thank you so much for being here, and let's start this journey. First disclaimer. You are going to see some images that are not going to be pretty. That's the reality of Leishmaniasis. So I'm going to alert you to when the lesions are starting to appear, just in case you want to close your eyes, but I hope you don't, because I want you to feel the reality of patients going through this infectious disease. All All right. Let's begin by one of the main definitions that brings us today together here, and one of the main aspects of validates REMIT, and is to work on neglected infectious diseases. So neglected infectious diseases, and in this case neglected tropical diseases, are diseases that affect a huge proportion of individuals within the world. Actually, 1 in every 8 people throughout the world is affected by at least one neglected tropical disease. That means 1 in every 8 individuals. There is 1 billion people that suffer at least one NTD. And you can see in this image, it is depicted as the orange people, but you can see also a blue person in the middle of the image as well. This is intended to show some other diseases that most likely you have heard of, like psoriasis, for example. It's a disease that is more commonly known and is more common to us because it usually happens in high-income countries, and we're more aware of these diseases. Psoriasis is not an infectious disease, but it's a disease of the skin. And so what I want to make you aware of is that these neglected infectious diseases affect a very huge amount of people throughout the world, but still we don't know very much about them. And we, as the general audience and people living in this world, have to be aware of this. So Leishmaniasis is a group of diseases caused by a parasite, and the parasite is called Leishmania. So it has 3 main clinical forms. The first is called visceral leishmaniasis. Essentially is the disease that is manifested by infection in the liver and in the spleen, and you have a lot of inflammation in this abdominal area, and this manifestation is deadly if it's not treated. It happens mostly in Africa and Asia, and we get around 50,000 people infected with visceral leishmaniasis. Then we have a second different manifestation, which is called mucosal or mucocutaneous leishmaniasis. So as you can see in these images here, it involves a very strong inflammation of the nasal mucosa, the nasopharynx, and it can generate so much tissue damage that you can see people losing the nasal septum, as you can see in the pictures here. This is irreversible. So if you get this advanced levels of disease, then you can have complications for life. And then the third different clinical manifestation is what we're going to be focusing today, which is cutaneous leishmaniasis. So cutaneous leishmaniasis is a disease caused again by the Leishmania parasite, but it causes lesions in the skin. And usually the lesions are in places that are exposed. And why is that? Because this is a disease transmitted by the bite of a fly, and we're going to see that in a minute. And so, you think, okay, it's just a lesion in the skin, it's just an ulcer, it's no big deal, it's going to be fine. But just imagine yourself, you're in the middle of the jungle, like these images here. This is in the Amazon jungle in Colombia, and you see 2 daily situations of people living in these areas. On the left-hand side, we see a person just harvesting their regular crops for a daily food. And then on the right-hand side, a person harvesting ants. I don't know if, has some of you guys tasted ants before? How was it? Not so nice? All right. Yeah, they're not that great, but they're very good in protein content. They're very nutritious. They taste a little bit like peanuts. But this is something that you commonly eat in the jungle. It's common practice. But what is common among these people is that they wear rubber boots, and this is the way that you have to walk in all these rural areas. And that's because there are additional dangers that you usually don't think of, like snakes, for example. And so if you are an investigator that go to these areas, or you're a person that lives in these areas, you always have to be very careful and walk with this type of boots. What happens if you have this type of lesion just in your knee? Just exactly where the boot hits the leg. And this is exactly the first case of leishmaniasis that I saw in my life. So I didn't mention to you, I am a microbiologist, I am not a physician. So I usually don't see patients. It's the physicians, the ones that see the patients. But the patients is what motivates me. And that's why I'm also in the clinic. And so when I did my first field trip to one of these areas in Colombia, I saw this guy. He was tall, like a very robust person. And it was incredible to see his teary eyes saying that he had not been able to fetch food for the family for 20 days. And that's when you start to understand what are the costs of this infectious disease. This is a disease that does not kill you, but has a very strong social impact and economic impact on people and families that are affected by this. And then there is another group of affected individuals that are children. And I'm not bringing these images to make your heart shrink, but to show you the reality of what Leishmaniasis is. And if you see, the commonality in most of these images is that children get the lesions in the face. Why is that? Because the fly that transmits the parasite has a flying altitude of about 1 meter. That means that if you're very small, you get bitten most often in the face, and that's why most of the lesions are in the face in children. There are some cultures where having scars in the face make girls not unavailable, but not desirable for marriage, for example. And this is just one example of the stigmas that Leishmaniasis can cause. In Colombia, Leishmaniasis has been called the disease of the war because it is transmitted in the Amazon area and in the forest areas where most of the conflict has occurred between the guerrillas and the armed conflict. And so a lot of people that go to seek medical care are very concerned that they're going to be considered a person involved in the war. So these are just some social stigmas that I wanted to share with you because there's so much more than just a skin lesion. And this is really what motivates us. And I come from a research center in Colombia. We are few. We are a group of about 50 people, but we have been doing research for about 60 years in neglected tropical diseases. And for many years, we have been working Leishmaniasis. And this is the most recent picture of our team involving, most of the research team is here, but some admin as well. And essentially, we're devoted to conducting research so that we can provide better healthcare for people. We do research from basic science all the way to applied science, meaning that we test drugs, we test interventions, and we also try to promote the appropriation of this knowledge in the community and within the community. And that's part of the reason why I'm here, because I want you guys to know about these diseases, to know about the work that is being done to treat these infections, and for you guys as well to spread the word that this is needed. In addition, we are a regional training center for the TDR. The TDR is a part of the WHO, but essentially we're committed to conduct research and training and capacity building in everything that has to do with neglected infectious diseases and training in the research itself. Becoming an investigator is not simple. And you think you do your PhD and then yes, you can do research. It's so much more than that. Anyway, we're going to get that in a minute. All right, so why did I decide to come from sunny Colombia to rainy UK? Although I was speaking with my colleague, he's like, yeah, it's not rainy today. So the joke did not work. But that means that I brought the sun to you guys. So I came here because I have had for many years a very strong collaboration with people in the UK. Just standing here and looking actually at the books in the library makes me feel amazing that this is a country that has supported science for so many years. This is a pillar for growth of humanity. And I'm really, really honored to be here talking to you about what we do. And so a lot of my colleagues are in London, some are in Glasgow, but it has been a very beautiful collaboration, and I want to give a little bit back as well to you guys. So, what is the Leishmania parasite? That is the bug that brings us today here. It is something that we call a protozoan parasite. So, I guess a lot of you have heard of malaria, for example. Malaria is also caused by a protozoan parasite, similar to the one that we're talking today. Leishmania is the purple one. And has anyone heard of the Moctezuma curse? Yes? Okay. So that's caused also by a protozoan parasite called Giardia. And it's essentially it causes really bad diarrhea, and that's the Moctezuma curse. But what is a parasite, or what is a protozoan parasite? It's a microorganism. We cannot see it with our own eyes. We need a microscope to be able to see it. But it's a microscopic organism that is much bigger than a virus. Bigger, like virus, like, for example, the one that causes COVID. Bigger than a bacteria. Bigger than a yeast, but not bigger than a mold. So we are in the side of eukaryotes, so cells that are similar to ours. But again, these are just organisms that are one single cell. We are composed of millions and millions of cells. This is just one tiny little cell that can cause a lot of damage. And I was saying at the beginning that this is transmitted through the bite of a fly. So we're usually used to the big, black, nasty fly. This is actually a tiny little fly, and if you see in the image, that is a notebook line. So you can imagine more or less the size of the fly. And if you can see, the fly is white. So it's very difficult to see. So when you're in the endemic areas where this infection happens, it's very difficult to find, it's very difficult for you to actually see the sandfly or the fly that transmits it. And so that's why we're usually not that very careful in those areas. So I caution you, Leishmaniasis is usually moving towards the north. So now we have endemic countries that we usually travel, or that you may travel a lot more, like Spain, Italy, now the United States. In the south part is also endemic for Leishmaniasis. So be cautious about this infectious disease because it's getting much closer to where we are. And these are the parasites. So essentially, the parasite lives within the gut of the fly. And once in the gut of the fly, it moves into the mouth parts of the fly. And once the fly bites us, it actually introduces the parasite that starts making the initial interaction with our cells. And I remember when I started my PhD, I did my PhD in Canada, but then I returned back to Colombia. My boss at the time used to say that Leishmania was like Medusa. And that's because the parasite is able to block every single, or not every, most of the functions that make our immune cells work. So let me just step a little bit behind. The cells in our body that are involved with eliminating pathogens or microorganisms are the cells of what we call the innate immune system. And you may have heard of them in your blood test: monocytes, neutrophils, macrophages. So these are the primary cells that go to the sites of infection and are supposed to be eliminating all the microorganisms from our body. However, the Leishmania parasite, and also, for example, Mycobacterium tuberculosis, the one that causes TB, lives within the macrophages, the cells that are supposed to prevent infection from taking over. And so that's why my boss used to call it Medusa, because it actually blocks all the functions of the macrophage, or these innate cells, so that it can survive, multiply, and perpetuate within the host. And the fact that it lives within these cells makes the development of drugs and vaccines especially challenging, because you can imagine that targeting a parasite or a bug that lives within our own cells becomes extremely complicated. Just imagine you being a drug. You have to go through your bloodstream, and then you have to go to the skin, and within the skin you have to go into the cell, and within the cell target the parasite. That is technically very complicated. And so it's a big challenge to develop drugs and vaccines for this, what we call intracellular pathogens. However, this is exactly what we are doing, and this is why we devote our careers to studying these infectious diseases. So again, what I want to bring you through today is the paths of patients and investigators working with neglected tropical diseases. And I'm going to start a little bit selfishly with my path, because I want to share with you the ups and downs of my life as a biomedical investigator. So I started as a microbiologist. I did my BSc in microbiology. And when I finished my degree, I was like, oh my God, I'm going to be working with viruses. Especially, I want to work with Ebola. So at the time that I finished my PhD, this movie came out. Has someone seen it before? It's amazing, huh? Yeah, so to me, I was just like Dustin Hoffman. That's what I wanted to be. That person that goes into the field and that also works in the lab for these very complicated infectious diseases. But the reality is that I ended up working with parasites. But I didn't want to work with parasites from the beginning. And why was that? Because I remember the classes that I took in my university. The classes were packed with complicated life cycles, like the parasite goes in the fly, and then it goes into the snail, and then it goes into the water, and I had to remember all of that, and it was really, really, really boring. And then you start seeing in Discovery Channel or in TV things like, you know, monster in me, flesh-eating bug, it's going to eat you. It's like, really, like, not that appealing. And so I would imagine myself much better working with hemorrhagic fevers than working with this type of parasite. But then, and this becomes a little bit, then I met this guy. He was my first mentor. His name is John Walker. He's no longer with us, but he's the person that gave me the passion to work with Leishmaniasis. And he shared with me what is to do amazing, beautiful science with a purpose. And so, thank you, John, wherever you are. All right. So, after I met him, I knew that I wanted to do a PhD, but I thought that I wanted to do a PhD in the usual beautiful places, and the usual beautiful places that I thought at the moment that I wanted to be was San Francisco, because at the time I was obsessed with Janis Joplin. And so I just wanted to be there, and I wanted to live in San Francisco. And then, of course, you know, things don't go as planned. I ended up in this beautiful place called Montreal. So imagine this person coming from the tropics arriving to that. That's actually a picture from when I was there. It was tough, but it was an amazing experience. And I'm bringing this picture because this is my second mentor. He was my PhD thesis supervisor, Martin Olivier. And when I first arrived to the lab, like literally the day after I got from Colombia, I went to the lab and he said, okay, I'm going to give you your PhD project. That was my PhD project. It's like, seriously? And so I went out of the room with a tear in my eye. Unfortunately, there was another colleague there that said, no worries, come on. I know what actually, he had different colors of sheets. I know what the green sheet is for, so I'll explain to you. So this was a beautiful journey, but at this stage, I knew about Leishmaniasis, but I had never seen the reality of Leishmaniasis before. And believe it or not, I have lived in Colombia. I mean, I am now 46 years old. I lived in Colombia before going to Montreal for like 25 years, and I had never seen a case of Leishmaniasis, and I had never heard of it before either. And so when I went back to Colombia for my PhD, then reality check, this is what the disease is. And it was even more motivating for me to continue working in this line of research. So Blakely was saying that I look really, really nerd in this picture. I don't know why. I mean, it doesn't look that nerd. But this is the first field trip that I ever did, and this was in Tumaco, one of the sites in the Pacific coast of Colombia. And that's one of the places where we have an outpatient clinic where a lot of people with Leishmaniasis go to seek care. And this is a picture showing the initiation of one of our clinical studies in the area. And I can show you a lot of pictures of this beautiful place, just some that you can see here so that you can go one day and visit this area. But in contrast with the beautiful nature, you start to see the reality of people living in these areas. And the reality is that there is a lot of poverty. And one thing that I didn't mention when I explained to you about neglected tropical diseases is that these are diseases of poverty. And they sustain something that we call the cycle of poverty and disease. Essentially, you have a disease because it is very difficult to get access to healthcare, both because there's not a lot of economic capacity, but also because you are living in very dispersed areas. You wait and wait and wait until disease becomes usually more severe. And then once you go and seek healthcare, it's very difficult to treat. And therefore, you sustain disease. And the fact that you have disease impedes you in many ways to sustain the daily working activities, and therefore it closes the cycle of poverty and disease. And so this is what I started understanding when I actually went to the field sites. So I really encourage you, if there are biomedical trainees or investigators here, you have to go to the endemic site so that you can really understand what we're working for. But this is not unique to Colombia. And this image I wanted to show you because I actually thought, this is a new slide that I'm using, and I thought it was really amazing. This is a map that shows the roads in the world. So you can see a big black spot that is in the Amazon region, just in South America, but it goes all the way to the tropical areas of Africa as well. That means that those are areas that have very little to no road connections. Just imagine you living in a place like this. That means that it is very difficult to access healthcare, amongst many other things. And this is precisely where these infectious diseases hit the most, in areas that have very little connection, that have low economic resources, but that are ecologically very beautiful. And why it becomes so complicated? That's because people or patients usually have to take many, many hours to be able to get from one place to the next. So this is an image, for example, of the north area of Colombia, Sierra Nevada, Santa Marta, where a lot of indigenous communities live. And this was us going to see a community that is 4 hours away of the main health post. You cannot imagine, I mean, I don't do a lot of exercise, but I do some exercise. And it was so tough getting up there. Just imagine a patient having to go up and down the mountain just to get diagnosed, and then up and down the mountain every day to get treatment. It's really complicated. So it's extremely time-consuming. And in addition, it can be extremely expensive. So this is another image of the Amazon area of the country, where I am showing you the typical transportation method, and that is through the rivers. So this is a number that is real. Patients have to pay around 20 pounds just one way to be able to get from their site to the main health post. Imagine 40 pounds on a daily basis when someone does not even earn the minimum wage, which in Colombia is 401 pounds a month. It strains any family. It's really, really tough. And what ends up with this is that people end up consulting to the clinic or to the health post very, very late, and leads to the severity of disease. So there are some images that are coming just now, and bear with me, but again, this is the reality of what we see. Patients seeking care too late. This is, again, another image of one of the health posts in the northern part of the country, and this is exactly what you see. We are waiting there for hours and hours and hours to see if someone is going to come, but it just doesn't happen a lot because people take too long to be able to arrive here. So we have, as an outcome, very severe types of lesions. And they can be lesions like this one. They can be cases where you have multiple lesions in your body. And it can be lesions that are as complicated as the one that you're seeing here. Actually, I'm very happy to have David. David used to be a physician and fellow in Colombia at CIDEIM, in the institution where we work, and now he's doing his PhD here. And he actually was the physician that took care of this patient. And cases like these are the cases that we are working for. So, I guess very appropriately, I discovered this image here. And if you have read your Agatha Christie, you may have heard about the main poison that Agatha Christie uses in the novels, which is arsenic. Okay, so we can relate with arsenic, and we can imagine that it's really, really poisonous. So just imagine if the drug that you're going to be using to treat Leishmaniasis is the cousin of arsenic, which is exactly the one. It's antimony. So this on the right-hand side is part of our elements table. And when the elements are very close by each other in this table, that means that they have properties that are very similar to each other. So if you see arsenic is AS and antimony is SB, and they are very close together. That means that the toxic properties of arsenic are very similar to the toxic properties of antimony. But antimony is the drug. So just imagine that you have a lesion in your arm and you need to have treatment with this type of drugs for 20 consecutive days, every single day. For a person more or less my size, I will get 7 milliliters of the drug in one butt and 7 milliliters in the other. Imagine what happens with children. We have to see people, very healthy otherwise individuals that have small lesions, having to receive the injections in the legs because they cannot support the injections in the butt. It's not a drug that we want to have at all, but this is the main drug that is still used for Leishmaniasis. And in addition of a drug being extremely toxic, it is a drug that doesn't work that well. In fact, 3 of every 10 people that are affected with Leishmaniasis will not heal, even if they have the perfect therapeutic scheme given to them. They will just not heal, and that's something that we call therapeutic failure. And in our group, we have been very interested in understanding why this happens, why there are some people that heal, but some others that don't. And by understanding those reasons, then maybe we can reorganize therapies so that they become more efficacious for the majority of the population. And so these are some of the challenges that we're looking to address with our research in Leishmaniasis in Colombia. First, facilitating access to diagnosis and treatment. So because of the distances and the difficulty in access to diagnosis and treatment, one of the most feasible ways to get healthcare closer to the patient is through mobile apps, through mHealth. And so one of the things that we work in our institution with is to develop applications for your cell phone, well, well, for the cell phones of the patients and communities, so that community leaders, for example, are able to identify or see a lesion and have more certainty that this is a leishmaniasis lesion and not, for example, an ulcer caused by a cancer of some sort or any other type of lesion. And that reduces the time the patient uses to seek healthcare, because he or she are more targeted towards what's needed. And on the other side, the other thing that we are very interested in doing is bringing healthcare closer to communities by bringing communities to be part of the healthcare. So in this example that I'm showing you here is a training activity that we're doing with community leaders in some of these endemic areas, where we teach them how to take a sample from the skin so that the patient doesn't have to move the 4 hours or 8 hours or 10 hours to the nearest health post, but the community leader can actually take the sample and then send it to the health post, and that actually makes the patient receive the diagnosis in a way that it doesn't require the investment of money and also the investment of time. And what's very dear to my heart is this part of basic science that binds the needs of people affected by these infectious diseases, but also by the beauty of the science itself. And so one of the things that we're very interested in is to identify biomarkers that allow us to predict whether a patient is going to cure or not if we give it the drug, so that people don't have to be exposed to these very toxic drugs unnecessarily. But what is a biomarker? So a biomarker is any molecule in our body, let's call it a protein, a carbohydrate, or a sugar, or a lipid, that correlates with a pathology of interest or with an outcome in our body of interest. And as I was saying, biomarkers come in different flavors. And what I'm going to try and do here is to try and to explain to you the types of biomarkers that we're working with. So I think we all agree that this is a beautiful caterpillar. I mean, it's pretty. But who would have thought that this caterpillar transforms into this butterfly? This is the blue morpho. This is very common in our country. Just imagine, this caterpillar has exactly the same DNA as the butterfly. So that leads us to what we call in biology the dogma of biology. That means that we start from our DNA, which are the building blocks of our genetic material. The DNA is the same in the caterpillar and the same in the butterfly. But then the DNA transforms into RNA. And the RNA then starts becoming different. And that's when we start selecting for specific genes to be expressed in one individual or another. And then those genes produce a protein, and proteins are also different between different life stages, but also between different individuals. And then we have finally the metabolites, which are things like glucose or for example, HDL or LDL that you know very well from your blood tests as well. So when we talk about biomarkers, it can be any type of molecule from here. But if we want to look for biomarkers that actually we can test easily in populations in endemic areas, the most easily evaluated biomarkers are those that contain proteins or metabolites, because we can do them with tools that are more easily implementable in these areas. And that's where we're focusing our work predominantly. So I'm going to go very quickly through this slide, but I just want to give you vision of what research from discovery to product means. And this is the funnel or the pipeline usually of product development. So essentially you start with a lot of different ideas, a lot of different products in the amounts of the thousands. And then you have a lot of hypotheses that you're testing, and this takes years and years and years, and it can take from 5 years to 10 years. And then you start funneling down, filtering and filtering and filtering until you get to less molecules that you can bring into preclinical studies. What are those preclinical studies? To evaluate whether our molecules, our drugs, our vaccines, our diagnostics are actually usable in living organisms. And we do this usually in animal models or in lab models. And then we go to the clinical research. And clinical research is most of the type of research that we do. That means that we go straight into patients or the affected communities to see whether these drugs or vaccines are actually useful for the pathologies that we're interested in. And again, these are rounds of years and years and years of research trying to narrow down what is the product that we're going to finally put into application for the communities. And so I just wanted to bring you 2 examples of where we are at this moment with this type of work. So in the biomarker aspect, what I was saying is that we are very interested in identifying those biomarkers that can allow us to know whether if you're treated with antimonial drugs, you're going to respond or not, so that you are not exposed to this very toxic drug. So in this stage, we're at the clinical research stage. So we're already analyzing biomarkers in human populations, and we have a set of biomarkers that we want to bring to clinical trials for validation to see whether we can put this forward to be used for patients. And we're looking at this in 2 different sites based on RNA and based on metabolites. And then we have a second big aspect of our research that is involved with drug development, and is to develop drugs that promote faster healing. So one thing that I didn't mention is that when you have these lesions, you have the 20 consecutive days of treatment, but it takes almost a full month to start seeing the recovery. So the idea is that we can use drugs that limit that time to recovery so that patients don't have to be exposed to the toxic drugs for so long. And so to do this, one idea that we have is that we can play with the immune system in a way that it favors the elimination of the parasite from the body and therefore promotes faster healing of these lesions. And that is also at the clinical research stage level. And then finally, the one that I wanted to bring your attention to is the most beautiful, I think, well, I'm biased, but one of the most beautiful experiments of nature, which is asymptomatic infections. And it's a beautiful experiment on vaccinology, I would say. So I think all of us, because of the pandemic, are now aware what an asymptomatic infection is. And for those of you who are not, asymptomatic infection is essentially when you get the bug in your body, but you never manifest any symptoms of disease. So you have been infected, but you don't manifest disease. And for this and many other infectious diseases, asymptomatic infection is a very high proportion of individuals infected with the parasite. So for example, if you go to an endemic area, for 1 patient that has a cutaneous lesion, 10 will have asymptomatic infections. These are people that have been infected, but that are protected from disease. So if we understand what are the bases by which these individuals naturally are protected from disease, not from infection, but from disease, we can definitely create vaccines that would be useful for future generations. And then I wanted to close this talk with some of the work that I never thought I was going to be exposed to, and that I cannot be more happy to have been. And that's the intercultural work that that it's not that we do, that we share with the communities. So if you go to different areas of Colombia, you will find a lot of different indigenous communities. And each indigenous community has a very specific vision of the world and also vision of health. And I'm bringing these examples because these are all communities that are affected by cutaneous leishmaniasis, but none of them think that this is a disease caused by a parasite that is transmitted by the bite of a fly. On the top left, we have the Awa community. They think that leishmaniasis is a disease that gets into your body because your soul is open and available for the disease once you pass through a bush, which is called the guaral bush. This is how they think leishmaniasis is transmitted. In the bottom, we have communities in the north of Colombia as well. They think that leishmaniasis is transmitted by the bite of a snake. And so understanding the vision of these communities towards the infection and the disease, you can get a more closer relationship on how to introduce the mechanisms to control these infections and to protect the communities from exposure to these parasitic infections. And that makes as well for beautiful and amazing travels. So when I asked you about the ants, I mean, I don't think that I would have even tasted an ant if it wasn't for the type of work that we do. So in all these different visits, we have been so blessed to be able to collaborate and work with the people that are just, it's just, I don't even know how to transmit this to you, but that's what brings us like the spark to continue doing this work. If you see in the image where we're having lunch, that guy in the right-hand side is called Carlos Ampudia. He's one of the community leaders that has worked with us for more than 15 years. This guy is not trained for medical care. He's not even an assistant of a doctor. And he's the person that is able to identify all the leishmaniasis patients in one of the communities that we work with. So I told you at the beginning that you were going to see very nasty pictures. And this is exactly what we work for. This is the patient that David treated, the patient with a whole bunch of lesions that you saw in the face is him. 3 of these kids are the kids that I showed you that had lesions in the face. They were part of some of our clinical studies. And this is what moves us to continue doing the work that we do. And so I hope that with this, you get some understanding and awareness of why we dedicate our days and nights and years to work with infectious diseases that don't have a lot of voice, because we end up being the voice of these patients. So with this, I'll leave you. I cannot, of course, finish without thanking a beautiful team of people that I have worked with for several different years. These are some previous and past members of our research team are here. Some of the community leaders that we have worked with for a while, and some of the funding agencies that have supported us for years, and hopefully for years to come as well. Thank you so much.
Adelaida, thank you. That was absolutely fabulous, really inspiring. So I think what's going to happen now is a couple of chairs are going to appear, and you and I are going to sit down, and then I'm going to invite the audience to ask you some questions. So while we're waiting for the chairs, do you think of some questions? Adelaida.
Great, thank you.
You deserve the first seat.
This is beautiful. All of this is Colombia. Okay, you have to go. These are some of the typical areas. This is Guaviare, beautiful as well. Beautiful. You have to go and see this. It's beautiful. Beautiful. And this is the grandchild of Carlos Ampudia, the community leader. And you know why they had a lot of leishmaniasis in a way is because all these areas are full of coca plantations. That's also part of our reality. And they have started doing this transition between coca plantations and cacao plantations. And this is his grandchild around the cacao, how do you call them? The things, the fruit, the cacao fruit. Those things are the cacao fruits. And they have this entrepreneurship called Choco Pimienta. So if you ever see it there, it's them. Anyway.
Fantastic. Great. Adelaida, take a seat. And I'd like to open the floor to questions. Does anyone have any comments or questions for Adelaida? Yes, let's start here.
Thanks, Adelaida. That was so inspiring. And I was just wondering why it is that Leishmaniasis is still treated with antimony, that there isn't another drug that can kill the parasite. Is it because of the parasites themselves, or the fact that they're inside macrophages, or, because it's quite an old... It feels like quite an old-fashioned, in inverted commas treatment. Like when syphilis was treated similarly with metal, right? Mercury,
exactly,
wasn't it? So why antimony, and where is the drug discovery story for Leishmaniasis?
Right. Thank you for that question. So what I didn't mention is that besides antimony, there are other 2 or 3 drugs that are used to treat as well. I didn't mention them because they're not the primary drug still used. The primary drug is still antimony. The other drugs are still too toxic as well. So for example, one, which is miltefosine, which is amazing because it's oral. It's extremely expensive, so patients actually cannot get to that drug. But it causes toxicity in the fetus if a woman becomes pregnant. So it's very complicated to deal with in terms of having contraception at the same time as having the drug in this type of communities. Now, why are we with the same drugs and don't progress that much? There's really not a lot of investment. That's the reality. And I don't know if we're going to start this new cycle of lack of investment, lack of drug, lack of investment, lack of drugs. But the reality is that there are not a lot of economical resources that go to investigating these infectious diseases, and therefore the length that it takes to move forward in the pipeline is so much slower. There are some candidates, and Wellcome, for example, has been really pioneering in supporting research in neglected infectious diseases. And one of the drugs that is currently in the pipeline for research is one drug. It's part of a project funded by Wellcome. So there's something in the pipeline, but still not a lot. Thanks.
One thing that I was wondering about, has anyone thought about using like drones to do airdrops of things? If your journey takes 20 quid and 4 hours to get to something, could villagers not have I don't know, a bullseye on the top of one of their houses where you say, you know what, we're just going to drop stuff there and help you get it?
Totally. So amazing that you asked this as well, because this is one of the things that we were discussing in a workshop, I think 2 years ago. There is beautiful research in Madagascar to use drones for delivery of drugs. I think it was for TB, if I recall correctly, not for Leishmaniasis. Now, the problem with Leishmaniasis is that if you were to use drones, the problem is that this is an injectable drug. So if it was an oral drug, then maybe that would be a way to go.
Go train people in the village to how to do an injection and then get an airdrop?
No. So the mHealth tools are to do diagnostic and treatment follow-up. So the other thing that I didn't mention to you is that to be able to say that a person has healed, you have to wait 6 months after you initiate drug treatment, because it can take that long for it to heal. So we use mHealth so that the person does not go on month 1, month 2, month 3, month 4, but we rely on the picture that is much easier. Now, The reality is that some of these community leaders actually put the injections, but this is a big risk. And the risk is that because this drug is extremely toxic, it can cause cardiac arrest or liver failure. So, you take it with a huge risk if you're not under proper medical supervision. But it's a great idea.
Jenny, another question here. This side of the stairs.
Has the pathogen been, has it done the genome sequencing, and did that lead to any kind of further research topics or drug development or anything?
Right, thank you so much. Yes, so the genome of this parasite is available, and it has led to very interesting discoveries, and very interesting discoveries mostly on how the pathogen is able to block the functions of the immune system. In terms of drug discovery, it has helped quite a bit, but there is one caveat, and is that Leishmania is not just one single parasite. I mean, how can I say this? Is one species, one genera is one parasite, but it has different last names. So it can be Leishmania panamensis, Leishmania guyanensis, Leishmania donovani, and so on. And the species is associated with the different types of pathologies. So what happens is that the genomes of those different brothers and sisters is almost identical. But I just don't want to say the wrong number, because otherwise my colleagues will get really, really mad. But let's say it's almost identical, okay? But they cause very different diseases. Some visceral, some in the nose, some in the skin. Why? If the genome is almost identical. So what we think is happening is that what I showed you about the dogma of biology is not in the genome necessarily. It's maybe in the RNA or in the proteins where we're going to find those answers. And a lot of work has been done as well on those aspects. But it is much more difficult to do a proteomic study, for example, that looks at the proteins than a genomic study. Technically, it's very complicated. But yes, I mean, all that very basic biology and parasite biology is what will allow us to move forward. And actually, one of my dreams, although I'm not sure if we're going to be able to do it, is that in these individuals that have asymptomatic infection, I would like to know whether the parasite that lives within them is similar or the same as the one that actually causes disease. Because if we know which parasites specifically are able to infect but not cause disease, then we really have the potential for a vaccine.
Lots of hands suddenly. Okay, Raiko, and then...
Oh, thank you. Thank you.
Fantastic story, Adelaida. Thank you for that. My question is about immunity. So the people you've treated here and cured, can they get reinfected? And if they can, what's the implications of that for vaccine design or our immunity against this pathogen?
Yes, amazing question. While I'm saying this, I'm thinking this is like a beauty queen, amazing question. But no, amazing question, actually. There are some people that can be, it is not known if they are reinfected or if a latent infection reactivates. So what we know is that if a patient gets infected with Leishmania, whether they have asymptomatic or a symptomatic manifestation, the parasite most likely is going to stay with them for life in, let's say, 50% of the cases. The rate of people presenting 2 episodes of Leishmaniasis is low. So you can get about, I would say maybe a 5%, if not less, of people that can have a second disease, a second episode of Leishmaniasis. So the majority of individuals are protected once they have this primary infection. So that gives us hope that there is potential for a vaccine, definitely. Now, that's a tricky thing to discuss, because also it is well known that people with asymptomatic infection, many years after they have been infected, can develop at one point symptomatic disease, but also a very, very few proportion of them. And what we think goes on there is not necessarily that suddenly the parasite just became pathogenic, but on the contrary, that an imbalance in the immune system of the individual allows that bug to flourish. So for example, like the case of shingles. Shingles is, yeah, I don't want to get the name wrong in English, but you can get disease reactivation many years after.
Young man next to Richard.
Hi. You've talked a lot about asymptomatic infection. Obviously, the main transmission is through the fly, but can transmission occur from an asymptomatic person onwards, or is it just through the fly?
So that's the holy grail of Leishmaniasis research. And that's actually one of the holy grails of a lot of vector-borne infection disease research. It is, I don't think that there is a yes or no answer at the moment. There are some pieces of evidence that suggest that individuals with asymptomatic infection can transmit. And actually, this disease, which I didn't mention, is also observed in dogs. So dogs can have lesions like the ulcers that I showed you. And asymptomatic dogs are very well known to transmit. Asymptomatic humans, the work that has been done has been primarily with mathematical modeling more than proof of concept that actually the fly can take from the skin. But what I think really goes on is that when your immune system is changing over time, the parasite as well has different levels in your body over time. So maybe you can be a transmitter. Let's say you have an asymptomatic infection, maybe you can be a transmitter when that parasite load or when that amount of parasite is much higher in your body because you have some immunodeficiency or some aspect of the immune system that is not controlling very well the parasite. So it's an amazing question, and actually that is the central question for elimination of this infectious disease, which it's in the plans for elimination, so let's hope it happens.
Hello. I'm just wondering about are you seeing any familial susceptibilities to this? Does there appear to be a genetic susceptibility to getting...
Right, so there have been studies on genetic susceptibility, but not extremely conclusive in humans. So in mice, it is very clear. In mice, there is a genetic susceptibility to infection and disease. In humans, the studies that I recall at this moment were some done in Brazil and some done in India, and they have contradictory results, some positive, some negative. So it is, I mean, I wouldn't put my hand on fire for genetic susceptibility. You weren't seeing a trend, no. Sorry?
You weren't seeing a trend? No. I have one more question. Do you use allopurinol much for treating this?
No.
No.
In Colombia, it's not used a lot, or at all, I would say. Maybe if you have like almost no option whatsoever, then some of these antifungal drugs will be used to try and see whether that helps. But really, the efficacy of these drugs is not that great.
I'm a vet, so we used to use, for dogs that were coming over that were positive, the antimony first, and then follow them on pretty much permanently on allopurinol. Whether or not it was working or not, you wouldn't know, but the majority of them were fine and asymptomatic, so doing well.
Yeah, so in humans there have been some clinical studies with allopurinol, but again, not very conclusive, and also with some of the azoles, and that doesn't seem to work that well.
Thank you, brilliant presentation.
Thank you.
Thanks so much for such a beautiful, moving talk. Wonderful. It's not my field, science at all. I'm a sculpturist and a world of arts. The moment I'm suffering from, I don't know what sort of parasites, but I know they are. But my question is, what about the vaccine? Is it going to be for any parasites if you have them, or is it the parasites you're talking about?
So the research that we do is specifically for these parasites that we work with. So in this case, it's for Leishmaniasis, but the network that we're representing is a network that works with other organisms, like the one that causes tuberculosis, the one that causes lepra, and the one that causes myelioidosis, which is a bacterial infection.
Yeah, we're going to need it. Doctors here, they don't know what it's all about.
Yeah.
Okay, let's move. So I think we've got 2 more questions, and then we'll probably pull it to a close. So we've got, 3. Okay, 3 more questions. So Andy, Marina, and then gentleman in the grey below Andy. Andy first. There's some of the audience I know.
Hi, Adelaida, great talk. So my question is mostly about the vectors and vector control. So we hear a lot about vector control for things like malaria and for, I can't remember what it's, mosquitoes, for example. What role do you think vector control has for Leishmaniasis? And do you think it's if we control the sandfly problem, do we control Leishmaniasis, or is it do we need to have an approach that targets multiple factors?
So in principle, if you control the fly, you control the parasite, because there's no other mechanism of transmission besides the fly. The problem is that vector control strategies in forested areas are unsustainable. So you're not going to go through the jungle spraying. That's just not possible. Or doing interchange of vector populations, like what is done, for example, for dengue. That's just unsustainable. So maybe in some areas where vector control is can be more efficacious in urban transmission settings, like, for example, what happens in Southern Europe, in Italy, in Spain, in those areas, vector control may be more sustainable, but definitely in the Americas and in some parts of Asia and Africa, that's not sustainable at all.
Mariana, Jenny.
Thanks. We have different approaches to tackle the disease, of course, and ideally all of them would work. One of them is developing a vaccine. Yeah. And say we manage to get to the point where we have a very effective vaccine that is long-lasting, we hit the jackpot. But then the other challenge is the reality of reaching the communities to get vaccination coverage. What is currently the vaccination coverage for regular vaccines with these communities?
So that depends on the infectious disease that you're dealing with. So serendipitously, I started working with yellow fever 2 years ago. So actually, my dream of working with hemorrhagic fevers became true 2 years ago. And for these type of diseases that are very severe, in the sense that they have a death rate that is very, very high, vaccination coverage is pretty good because there is a lot of awareness within the communities that there is need. Now, that is a different story if you go to the urban areas. If you go to the capital city, Bogota, for example, vaccination coverage for yellow fever is like 0. I mean, not zero, okay? Don't take me that. But it's going to be very, very low, and that's because people don't think that it's necessary to have that vaccine. So I think one of the main aspects of vaccine research, besides let's say the immunology or the biomedical research itself, is the social science that comes as well with vaccine development and vaccine deployment, because there has been so much disinformation, unfortunately, because of COVID. But I think that there's an opportunity. I mean, as far as there is a clear understanding that this is meant for protection of human health, then in that case it should be, I mean, my hope is that in that case it should be okay. So the experience that we have for yellow fever is supportive of willingness to receive vaccination, at least in Colombia.
Okay, last question. Gentleman in grey behind you, lately.
Thank you. Really interesting talk. I knew nothing about any of this before I came here. I think you've actually just largely answered my question, which was about the whole disinformation thing we experienced, particularly around the COVID vaccines, and whether that was likely to affect the communities that you're particularly working with.
So indeed, yes, it brought misinformation to everyone. It's incredible how that traveled. I mean, you cannot get diagnosed, but you can get all the information or misinformation about the COVID vaccine in the Amazon. It's like unbelievable. But there is always, I mean, if you have a relationship that is based on trust with the community, then you can really get things happening. And I think that that's one of the most important aspects of the work that we do, is besides the molecule, you know, is building the trust so that the work that we do can actually be implemented. And now there is a huge movement of not bringing the science that we think is important for the community, but actually bringing the community to decide what is important for them. And actually, one of the most beautiful things when, you know, the first time that I went to one of these field visits, I was very naive, and we did a very non-structured survey about what the community thought it was relevant for them to, for us to do research in their context of what we could do. And consistently, the answer is education, education, education. I mean, I was very surprised that they didn't say a new drug, a vaccine, is education. So it all starts there. If you're able to build those relationships of trust where you can promote education, and then through that convey the need and the appropriateness of the control strategies that are based on biomedicine, then we're going to have the way to go. That's what I think.
Thank you very much.
Sure.
Great, that's a great way to end. So I'm going to draw it to a close, but Adelaida is still around, so when we've closed, do please come down and chat to her. I think she has inexhaustible energy to talk about these things. So, and I think you'll agree she's given us a fabulous talk. If anyone would like to know any more about Validate, please catch Blakely or Jenny or someone with a lanyard. And thank you all for coming and for making this such a special evening. And I'd like us all to put our hands together and thank Adelaida again, please. Thank you.